In this video, we are going to take a look at Goal 3 for our North Carolina Biology Standard. So this is going to cover pretty much all of genetics and evolution, touching on DNA, so the structure and function of DNA, protein synthesis, genetics, so Mendelian and non-Mendelian inheritance. We're also going to talk about biotechnology, and again, evolution, natural selection, and then as well as the classification system as well. So this is going to be a long one. Okay. So starting off talking about DNA, remember our structure of DNA is described as a double helix or a twisted ladder. So it is a double helix. So you have the two outside strands of the double helix, which are your alternating phosphate and sugar groups. The rungs of the double helix are going to be your complementary nitrogen base pairs. So your base pairs are always adenine and thymine that are paired together, and then cytosine and guanine that are paired together. So if we look at the picture here, we say, right, yellow and red, right, always being paired together, yellow and red, and then green and blue always being paired together. Now these base pairs are held together by hydrogen bonds. Those bonds are pretty weak. Think of hydrogen bonds as the equivalent to Velcro. It's sturdy, but it can be easily pulled apart. Now the sequence or the order of the nucleotides within our double helix is going to be the code for proteins. So if you think of, right, like a book, right, that you would read. So in the book of humans, right, there would be 46 chapters because we have 46 chromosomes. So every chapter is like a chromosome. Well, within each chapter, you have paragraphs, and then each paragraph is made up of sentences, okay, which are then made up of letters. You with me so far? So if the chapter is a chromosome, the paragraph, okay, the sentences would be a gene, and then each letter would be one of our nucleotides of DNA, A, T, C, or G. So these sentences, they are the genes that code for proteins. And that is a key, right, function for all life, for all cells. We do need to replicate DNA whenever we are going through cell division, so either mitosis or meiosis. Replication of DNA does happen during the S phase of the cell cycle, and it is what allows the daughter cells to have an exact copy of the parent DNA. Cells can respond to their environment by producing different types and amounts of proteins, but remember, with very few exceptions, okay, all of the cells within an organism are going to have the same DNA. What gives us different types of cells, a heart cell versus a lung cell, is the expression of the genes, right? Is the production of the different proteins, okay? So again, looking at our sequence, right, or not our sequence, our structure of DNA, right, we can see it is a double helix. So we have, right, that twisted ladder, okay, compared with RNA. RNA is a single helix. Okay, it's still kind of twisted, but it's only a single helix. And then we have our four base pairs, G, A, C, and T. And RNA also has four base pairs, but the exception here is uracil, right? The other three are all the same, A, C, and G. But RNA does not have thymine. Instead, it has uracil. So the advantages of right, overproduction of proteins, right, at the incorrect time, right, you can repair right, damage to cells. The disadvantage, right, to having the wrong number of proteins is cancer, right, which is when a cell is dividing uncontrollably, or when a cell is just dividing when it's not supposed to be dividing. Protein synthesis is the process of making proteins. If you synthesize something, that means to make. So we are making proteins. The process of protein synthesis occurs in two stages. Step one is transcription. Okay, so if we see here like script, like to write, and then trans means to change. So transcription is when we are changing DNA into RNA. So transcription produces an RNA copy of DNA. Okay. This specifically, this is called mRNA. The M stands for messenger because it is carrying a message from DNA. Because remember, DNA is a double helix, and it's also going to contain every single gene. So again, if we think of DNA as like a chapter in a book, I don't necessarily want to copy the entire chapter, right? It may be 18 pages long. I only want a single gene. I only want a single paragraph within that chapter. That's what mRNA is. It is a copy of a single gene or a single paragraph from the entire chapter that is DNA, so chromosome 1. So now that we have rewritten DNA into mRNA, the mRNA is going to leave the nucleus and it's going to go find a ribosome. On the ribosome is where the second part of protein synthesis can occur, which is translation. Translation, right, like in real life, where we change from one language to another, so from English to Spanish. Same thing happens in protein synthesis. We are going to translate from the nucleic acid language, RNA, into the protein language. So translation, we are finally making a protein. Now, there is a second type of RNA called tRNA that supplies the amino acids to the ribosome. The ribosome then links the amino acids together using a peptide bond. And then we finally have our protein or our polypeptide. Polypeptide is just a fancy way of saying protein. Now, different proteins have different structures, therefore they have different functions. Okay? And it is the order of the amino acid sequence, right, that determines the function of that protein. So is it going to be hemoglobin and help carry oxygen, right, in our blood? Is it going to be myosin, which is a protein in our skin, right, that keeps our skin, elastic? So it's the order of the amino acids that determines the function of the protein. When we are doing translation during protein synthesis, we use what's called a codon chart to help us to determine the order of the amino acid sequence. So, for example, okay, what would DNA codon CCG be? Okay, so this is DNA right here, okay, CCG. So the first thing we have to do is we have to transcribe our DNA into RNA, okay? We're going to use our base pair rule. So C pairs with G, C pairs with G, G pairs with C, okay? So I just went here and I looked at my DNA and then I used my base pair rule. Now that I have my RNA, that I have done transcription, I can now do translation. So I'm going to use my codon chart here. Right, my first base is a G, so I know I'm going to be in this row. Okay, so G. My second base is another G. Okay, my second base is a G. So now I know I'm going to be in this column. Okay, so I see where it overlaps down here. My third base is a C, which I have drawn over, so I'm going to have to get rid of it. So my third base is a C, so GGC gives me the amino acid glycine. There we go. So that's how you would use a codon chart in order to find your amino acid sequence to build a protein. One thing I'd like to point out here is that we have multiple codons, so CCU, CCC, CCA, CCG, right? So we have 4 different codons that all give us the same amino acid. This is really important because when it comes to spelling mistakes or DNA mutations, not all of those mutations are seen in the phenotype of the organism because, right, even though you have a mutation, you still got the correct amino acid. So a lot of the times mutations are silent. So they happen, it's a spelling mistake, but there's no effect because you still got the correct amino acid. So I think that's just something that's really cool about transcription and translation, but is also very important for us to be able to understand when it comes to DNA mutations. Haha. So a DNA mutation is just simply a change in the DNA code. Okay, mutations are random. Okay, there is no way to like predict or make one happen. They are random. They can be caused, right, by radiation, exposure to a chemical, um, you know, so secondhand smoke, the mutagens. Okay, the mutagen just causes a DNA mutation. Now, a DNA mutation, we can delete a letter, we could add in an extra letter, or we could substitute or switch out a letter. Okay, all three of these are just different types of DNA mutations that can happen. Now, only mutations that occur in our sex cells, the egg or the sperm, are going to be able to be passed on to offspring. So if you have a mutation that occurs in a skin cell, right, and you develop, sorry, there it goes. So if you have a mutation that occurs in your skin cell, right, and you develop skin cancer, that you are not necessarily going to pass that on. Only a mutation in a sex cell can be passed on, right, to the next generation. Okay. Now, speaking of the next generation, right, meiosis. Okay, meiosis is the process of creating our gametes. Okay, we know from the work of Gregor Mendel and also technology today that genes are on separate chromosomes. So you have some genes on chromosome 1, chromosome 2, but because genes are separated onto chromosomes, this allows them to be shuffled, right, during meiosis. So just like I would shuffle a deck of cards, we can shuffle our genes. Okay. The process of meiosis, or the production of our sex cells, so the process of meiosis leads to an independent assortment, right, of all of the different genes, which then ultimately creates a lot of genetic variation or a lot of diversity, right, within that species or even just within that family. So genetic variation, right, in sexually reproducing organisms includes crossing over, right, or trading genes from one chromosome to the next, random assortment of chromosomes, like they're lined up randomly during meiosis, so you don't know, right, which version is going to go into which cell. You can also have random gene mutation, and then you can also have something that's called non-disjunction, and this is when a chromosome pair fails to separate properly. Non-disjunction can result in a lot of genetic disorders, such as Down syndrome, where you have 3 copies of chromosome 21. You're only supposed to have 2, but because of non-disjunction, you end up with 3. And then also fertilization, right, can create genetic variation, because again, you don't know which egg and which sperm are going to fertilize. So it's completely random. So there are a lot of sources of genetic variation when it comes to organisms that reproduce sexually. So again, that random assortment, right, meiosis, it's cell division that creates our egg and sperm cells. And the random assortment we're talking about occurs during metaphase 1. So again, those chromosomes, they line up randomly, which basically means that, right, we don't know which way they're going to get pulled. Okay, we don't know which gamete they're going to end up in ultimately. Here's a good graphic of meiosis. I do want to point out what's happened here, right? So we can see in this first cell where we just have 2 plain red chromosomes and 2 plain white chromosomes, but then we now have like this white chromosome that's got a little bit of red on it, this red chromosome that's got a little bit of white. That is crossing over. Okay, so those two chromosomes, they have exchanged genes. So this is crossing over. Okay. But then again, right at the end, right, our 4 daughter cells, they are all different from each other because of crossing over and independent assortment. So no gamete is identical to another. So comparing mitosis and meiosis, so mitosis is asexual reproduction because, right, there's only one parent that's involved. You do make 2 identical cells, and those cells are your body cells or somatic cells, so lung, liver, heart, spleen, all of that. You do have a diploid number of chromosomes, meaning you have a full set. So for humans, right, that would be 46 chromosomes, right, because it's diploid. The number is going to stay the same. Okay. Whereas in meiosis, this is sexual reproduction because you do need 2 parents, right? You need mom and you need dad. And then you also make 4 cells, and they will be genetically different from each other. Okay, they are gametes, so egg and sperm. And then those gametes are haploid, meaning they have half the number of chromosomes. So instead of having 46 chromosomes, each gamete would only have 23. Okay, because again, those 2 gametes, they're going to combine together during fertilization to get back up to 46. So we need to make sure that the sex cells or the gametes that are produced during meiosis have half the number. That way they can recombine to get us back up to our diploid number. So again, just kind of comparing mitosis and meiosis. So we can see up here in mitosis, right? See how we are starting with 4 chromosomes, and then we replicated the chromosomes, but we still only have 4 of them. We just went from having 4 single chromosomes to having 4 double chromosomes. Then we divide, and both daughter cells still have 4 chromosomes. So we didn't gain or lose anything. Whereas in meiosis, okay, so I start with 4 chromosomes. I still do DNA replication. Okay, so again, I just went from having 4 single chromosomes to 4 double chromosomes. So single chromosome versus a double chromosome, right? Then I go through the first round of meiosis, and I can see here I have 2 chromosomes in each daughter cell. Then I divide again, right? And so my 4 gametes that are made at the end of meiosis, right, all 4 of them only have 2 chromosomes. So I cut the number in half. I went from 4 to 2. Okay, so the number was cut in half. Because again, let's say that this is a male and these 4 daughter cells are going to become sperm. That sperm is going to fertilize an egg, 2 plus 2, to get me back to 4. So we're going to talk about a little bit more about genetics now. If you need to take a break, remember you can always just pause, come back. But now that we've got a little bit better of an understanding on DNA as well as sexual reproduction, we can talk about, right, how these traits are actually passed from parent to offspring. It's really interesting. We actually learned a lot more about genetics before we even knew DNA existed through the work of Gregor Mendel. And then his work was kind of lost to history. We kind of discovered DNA as a molecule, with Watson and Crick. And then we kind of rediscovered Gregor Mendel's work on genetics. So it kind of went back and forth a little bit. But one thing we need to be able to do, right, for our test is we need to make sure that we can determine parental and offspring genotypes and phenotypes, okay, depending on the type of inheritance. So the first type of inheritance we need to be familiar with is called codominance. So codominance is when traits are equally expressed, so both parents, so think codominance. So that would be like a red parent is crossed with a white parent, and then the heterozygous form is a roan cow. So that cow is both red and white, like what we see up here right in our picture. So that is the codominance. Both are being expressed. Incomplete dominance is when you have a blending of traits. So think about when you blend paint. If you take red and white paint and you mix it together, you get pink. So here your heterozygous form, big R, little r, would be pink because it is incomplete. The dominant trait is not completely dominant over the recessive trait, right? And then polygenic traits, right? Poly meaning many, genic referring to gene. So a polygenic trait is a trait that's controlled by more than one pairs of genes. Polygenic traits are quite common among humans. So height, hair color, eye color, skin color, all of these are polygenic. And so instead of just seeing, black or white, you see a range of traits, right? We're not just tall or short, right? We're everything in between. So for polygenic traits, right, it really is it's a spectrum, okay? So you have like, super recessive, super dominant, and then a lot of in between, okay? So eye color, again, height, skin color, all of those are polygenic for humans. A karyotype is, it's actually really fascinating. It is a picture, right, or a map that can help us to identify the sex of an individual, and it can help us to identify if they have a genetic disorder. So again, right, most humans, we are going to have, right, 46 chromosomes. And then if we are female, right, we're then going to have 2 X sex chromosomes. There we go. Sorry, it takes forever for me to write with this tool today. Or, right, if we are male, then we are going to have 1 X sex chromosome and then 1 Y sex chromosome. So we always look at the 23rd set of chromosomes to determine the sex of the individual. So 23rd is down here. It's really easy to tell if an individual is male or female because the Y chromosome is so tiny compared to the X chromosome. I mean, just look at how much smaller that Y chromosome is compared to the X. So if you see that discrepancy between size, it's X and Y, and therefore the individual is a male. With this karyotype, we can see that there is a disorder that has occurred right here at chromosome number 21, because we have 3 pairs of chromosome 21. And so this individual would have Down syndrome because they have an extra chromosome 21. Now, let's say this individual only had like 1 copy of chromosome 21. That would not be Down syndrome. Down syndrome is specifically 3 copies of chromosome 21, okay? If it's any other chromosome that you have extra or missing, it's going to have a different name. But Down syndrome is probably the one that you're going to need to be familiar with as far as our test is concerned. So you can use a Punnett square, right, to determine genotypic and phenotypic ratios. So what is the genotypic ratio of the square below? So when we're talking about genotype, we're talking about like the actual genes. So for homozygous dominant, that would be 0. There are no squares below that are homozygous dominant. Now, as far as heterozygous dominant, right, we have 2. So that would be, right, 2. And then for homozygous recessive, again, right, we have 2 offspring down here that are homozygous recessive. So that would be the genotypic ratio. As far as the phenotypic ratio, let me pull out another color here. The phenotypic ratio, right, we're looking at the color, so yellow versus green, and that would be similar. These 2 are going to be yellow because they do have that dominant allele. And then these two, because they don't have a dominant allele, they are going to be recessive. So this would be a ratio of 2 to 2, right? Which we could then reduce to 1 to 1. Okay. So that's how you would use a Punnett square to determine genotypic and phenotypic ratios. So we also need to be familiar with inheritance patterns. So an autosomal inheritance pattern, autosomal just simply means that you are on chromosome 1 through 22. So you are not on a sex chromosome, okay? So chromosome 10, whatever. Sickle cell anemia is an incomplete dominant recessive pattern, but again, it's on chromosome 12. I don't know that specifically. I'm just throwing out an autosomal chromosome. Okay, again, it's not sex linked. Okay, so sickle cell anemia, cystic fibrosis, Huntington's disease. These are just a couple of examples of autosomal inheritance patterns that they might give you a Punnett square, and you would need to determine, what is the likelihood that a child, would have cystic fibrosis if, and then they tell you the parents. One interesting thing, though, about sickle cell anemia is that individuals that are carriers for sickle cell, so they're not affected themselves, okay? They don't have any of the symptoms of sickle cell anemia, but they have the added bonus to their immune system of being much, much less likely to get malaria, which is just super, incredible, because again, having your genetics, that's outside your control. But if you are heterozygous for sickle cell, you not only don't have sickle cell, but you're also a little bit more slightly immune to malaria. Now, but here's the risk. Because if you're a carrier and you also marry a carrier, right, and have kids, right, you do run the likelihood of having a child that will get sickle cell anemia. Okay, so give and take there. And then this question over here: if a male with Huntington's marries a female without it, what would be the chances of their having a child having Huntington's disease? So Huntington's disease is dominant, which means I only have to have one copy in order to be affected. So if you have a male with Huntington's, he's probably going to be heterozygous. He could be homozygous dominant. But then he marries a female who does not have Huntington's. Okay, so what would be the likelihood of them having a child that does have Huntington's? So that would be at least 50% of them having a child that would also have Huntington's. So that's an example of like a type of question that you could see, about genetics and inheritance patterns. Blood types are really interesting because blood types, they are a really good example of codominance and multiple alleles. So remember, codominance is when you have both alleles being expressed at the same time. So that's people who are AB blood type, right? Because A is dominant and B is dominant. And it's also an example of a multiple allele because there are 3 alleles that determine blood type, right? You have A, B, and O. Okay, so those are your 3 alleles. So you might have to be able to set up a Punnett square for blood type. So just remember that you can be homozygous dominant for blood type A or heterozygous dominant for blood type A. Same thing for blood type B. Okay, you can have 2 big B's or you can have a big B and a little I, okay, which is the O. So if they ask, can a mom with blood type A and a dad with blood type B, can they have a baby with blood type O? The answer to that would be yes, right? Mom would have to be heterozygous for blood type A, and dad would also have to be heterozygous for blood type B. only in that situation would they be able to have a baby that would have blood type O. So it could happen. And then the next type of inheritance pattern is sex-linked. So we talked about autosomal disorders like Huntington's, which is on, like chromosome 2. Sex-linked disorders are on your sex chromosome, okay? They are recessive, okay? If it's sex-linked, it is recessive, okay? Are there dominant sex-linked traits? Yes. Do we care about them? No, okay? So if you see sex-linked, recessive. Now, males are much, much, much more likely to express a sex-linked trait because, right, males only have one X chromosome, right? Their other sex chromosome is a Y, right? And there's no allele on that Y chromosome because it is an X-linked trait, right? X-linked, sex-linked. So males are much, much more likely to express a sex-linked trait because they only have to have one copy versus females, which would still need 2 copies to be affected. Color blindness and hemophilia are 2 of the main sex-linked disorders, right, that you might be asked about, right? What's the likelihood of a child being colorblind if the parents are blah, blah, blah. But again, you're just setting up your Punnett square like normal, with the exception that you've got to keep track of the parent sex chromosome. So in this case, it would matter if dad is colorblind or if mom is colorblind because, right, dad's Y chromosome cannot carry a sex-linked trait. Only the Y chromosome can. And then we have pedigrees. Pedigrees are essentially family trees. Okay, they show the pattern of inheritance of a particular disorder within a family. Males are always going to be squares, females are circles, and if you are affected with the trait, then you will be shaded in. Okay, so in this pedigree, right, individual number 1, number 7, and number 7, 11, and 13, right, they are all affected with this trait. Now, if we were to try to figure out the type of inheritance, we do see that it does show up in every generation. But I do see that there are 2 males that are affected, and then 2 females. So this is probably going to be autosomal. Looks like autosomal dominant. I know it says pedigree with X-linked recessive inheritance, but that's just not what this is telling me. I don't think that's true. Because again, right, dad's got it, and then daughter's got it. So, and then this, I guess this grandma has it, and then she passed it to, right, son. So to me, this looks like it's autosomal because it's showing up in every generation. So the genotype of person 11, right, so this dad over here, he doesn't have it, so he would be recessive. I'm just going to pick the letter A because I feel like it. So he'd be homozygous recessive. So the only allele dad could pass to his son would be a lowercase a. But again, this son is affected, so he would have at least one uppercase A, which we know he got, right, from his mom, right, just above. So this would be autosomal autosomal dominant. So ignore that at the bottom. I don't think that's correct. And then finishing up talking about genetics. So we do need to understand the relationship between genetics and the environment. So quite literally the abiotic environment around us, like sunlight, or exposure to toxins or chemicals in the environment, and how those can affect our traits or our genes. But also environmental factors as in decisions that we make. So do you use tobacco, right? Because that could increase the likelihood of developing lung cancer. Are you constantly being exposed to the sun? Do you work outside? that can increase the likelihood of skin cancer. But both lung and skin cancer can also be genetic. So you can be genetically predisposed to having lung cancer, and then you use tobacco, and then that further increases your risk. So there are some traits that can be influenced by not only your genetics, but also by the environment or the decisions that you make. Do you have a good diet or access to good nutritional food? Because that could affect whether you develop diabetes or whether you're overweight or underweight. So there is a relationship between some genes and their expression, right, when it comes to the environment. However, there are some traits that are not affected, like blood type. No decision you make will ever affect your blood type. You're born with it. It's there. It will always be, O positive or whatever. So not all traits can be affected by the environment. So kind of moving out of genetics and into a little bit of our evolution, we're going to talk about DNA technologies or biotechnologies. The first type of biotechnology we need to be familiar with is gel electrophoresis. Gel electrophoresis is the process of creating a DNA fingerprint. Essentially, a forensic scientist will use a restriction enzyme, which is a special protein found in bacteria. We'll use these restriction enzymes to cut DNA into different sized fragments, and then we run those fragments through a gel. The gel is charged. That's kind of where that electro part comes in, right? Electrons or electricity. So we will run a current through the gel, right? Negative to positive, and DNA has a negative charge. And so, right, that's what pulls the DNA down to the bottom. The shorter the fragment, the farther it'll go. So to create a DNA fingerprint from a gel electrophoresis, it is the charge and the size of the DNA that causes it to move. Because again, DNA has a negative charge, right? And again, the smaller the fragment, the farther it'll move. So sticking with different biotechnologies, we're going to talk about transgenic organisms. So transgenic organism is any type of organism, plant, animal, bacteria, that has been, where its DNA has been manipulated. So essentially what we do is we take a gene from one organism and we put it into another organism. This has been done a lot in agriculture, pharmaceutical industries. Basically, one of the very first transgenic organisms we created was bacteria, where we inserted the human gene for insulin into bacteria, and then every time the bacteria reproduced, they would also create insulin, which we could then bottle up, right, to help treat people with, diabetes, which is fantastic. So transgenic just means, right, changing genes, right? And that process of creating a transgenic organism is called transformation, okay? Now, there are a lot of ethical issues around biotechnology. Not so much the whole DNA fingerprinting forensic science thing. That one's not very ethical. We're using it to catch, bad guys, using it for maternity or paternity testing. But when it comes to, gene therapy or cloning, stem cell use, even transgenic organisms, there are a lot of ethical issues just because, how do we know if there's any side effects? Who gets to decide which genes are cut out and put into other organisms? It's all great and everything that we're treating diabetes, but who's to say where we draw the line in what we can and can't do with these technologies? So that does bring up the ethical concern around a lot of gene therapy and other biotechnologies. So moving into getting close to the end, evidence of evolution. So remember, evolution is just simply the change in a population over time. And when we say time, we're talking about geologic time, generational time. So how a population changes from one generation to the next over millions of years. So beginning with evolution, right, we talked about, right, early Earth, how early Earth was not suitable for organisms. We talked about Miller and Urey, right? They did an experiment where they recreated Earth's early atmosphere, and they were able to create organic compounds such as amino acids. And if you'll recall, organic compounds, proteins, carbs, lipids, nucleic acids, right? Those are essential for living things. So Miller and Urey did show that the early Earth would be suitable for the formation of, right, life. So very first organisms on Earth, they were anaerobic prokaryotes, right? Your bacteria, okay? Then we got photosynthetic bacteria, okay? We got our chloroplast. Then we went on to eukaryotic organisms, then multicellular. So that's kind of the progression from, right, very first to what we see today, with animals and plants and everything being multicellular. So couple, right, pieces of evidence. We have fossils, right, which informs our understanding of how species have, again, changed over time. We can compare, right, ancestral fossils to either fossils found today or even living, descendants, right, how have they changed, how have they stayed the same. Biochemical evidence, again, looking at molecules, so either an amino acid sequence or a DNA sequence. This is fairly new technology, but it is very accurate, right? If we take a look at a DNA sample and we can compare it, right, to a currently living, bird, and then maybe a fossil that we found to see, are they closely or distantly related? We can also use anatomical structures or anatomy to tell us if organisms share common ancestors or not. You have homologous structures, and you also have analogous structures that can tell us whether they share ancestry or not. Homologous meaning that they do share a common ancestor. Analogous structures, they do not share a common ancestor. And then we have, right, natural selection, Darwin's theory of evolution by natural selection. So again, natural selection, right, kind of term survival of the fittest. You have a lot of variation, right, within a population, genetic variation, but also phenotype, right, what you physically look like. And it is because of that variation that nature, right, will favor or pick, right, whichever trait is best suited for that environment, and that is the organism that survives, reproduces, and passes that favorable trait on to the next generation. So then we would see that the next generation, right, there are more individuals with that favorable trait. So, this accumulation, right, and change in, right, the genes over time leads to evolution. Okay? It can also lead to what's called speciation. Speciation is the formation of a new species. One of the main driving factors of speciation is geographic isolation. So there is a physical barrier preventing, two populations of squirrels from interbreeding, and over time they just become, right, so different from each other that eventually they become two distinct species. So that is kind of natural selection, again, in a nutshell. Natural selection can also be observed in antibiotic resistance and pesticide resistance. So remember, when we talk about our immune system, right, you have your passive immune system, like vaccines or breast milk, and then you have active immune system, right, which is where you are actively, right, creating antibodies against the exposure of a pathogen like bacteria, viruses, or fungi. The only thing we have to treat viruses are antivirals and vaccines. Whereas if you have a bacterial infection, right, we can use antibiotics. So again, right, antibiotic resistance, pesticide resistance can lead to evolution, right? Natural selection. So you have, right before selection, you have all of this variation in your population, right? So various shades of red, orange, and yellow. But then you take an antibiotic, right, because you're sick, and you kill off all of the bacteria that, you kill off all the bacteria. But some of those bacteria survive because they happen to be immune to that antibiotic, right? So our final population, right, they look way different compared to their ancestor population. So this is natural selection, right? So we see a change in this population over time due to taking antibiotics. But we also see the same thing when it comes to pesticide resistance or spraying pesticides right on our plants, right? They develop an immunity, or they have an immunity, and then that's passed on right to the offspring, and now everyone's resistant. That sucks. Okay, getting closer there. So classification system is constantly changing, constantly. Back in Aristotle's days, whatever year that is, there were only 2 kingdoms, plant and animal. That was it. Today, we have 6 kingdoms. Okay, so we have greatly improved the number of kingdoms. Why? 2 reasons. We discovered new organisms, and we developed better technology. And that technology also helped us to discover new organisms like bacteria, because they're microscopic. I needed a microscope to see them. So new technology and discovering new organisms are the 2 reasons why, right, our classification system is constantly changing, okay, right, based on new knowledge. We have 3 domains and 6 kingdoms within our classification system. The 3 domains, archaea, bacteria, and eukarya, and then the 6 kingdoms are down here, which are the ones you do need to be a little bit more familiar with. And then we have kind of our current classification system, which is based off of Carl Linnaeus. So starting with, right, the largest level, which is domain, and then we have kingdom, so like the domain eukarya, and then you have the animal kingdom, and then phylum, class, order, family, genus, and then finally species. So humans, right, we are animals, we belong in the animal kingdom. But the closer you get to the species level, the more specific you are, whereas up right near the kingdom level, right, that's much more broad, because the animal kingdom includes all animals, so humans, lions, insects, all of the animals. So very, 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 very distantly related to each other. Also within the classification system, we have what are called dichotomous keys. Dichotomous keys help us to identify the names of organisms. So a dichotomous key, we use pairs of statements, and we use physical characteristics. Okay, you can't be subjective. You can't say, oh, it looks gross, or it's tall. That's subjective. Okay, so they want us to figure out the name of organism A. So I always have to start, right, with number 1 for each organism. So has pointed ears or has rounded ears? Well, I definitely think those are some pointy ears right there. So I am going to go to 3, okay? So 3, ears are pointed upwards or ears are pointed downwards. Those are definitely pointed upwards. So I'm going to go to 5. Engages in waving behavior or does not engage in waving behavior. It's not waving at me, so I'm going to go to 6. Has hair on head or has no hair. I definitely think that's hair on its head. So this would be the name of organism A. So I'm just going to put an A right there. All right, well, what about organism B? Let's see. So has pointed ears or rounded ears. I guess, yeah, I think those are still pointed. So I'm still going to go to 3. Those ears are pointed right upwards or downwards, definitely pointed downwards. So I'm going to go to 4. Engages in waving behavior, has hairy tufts on ears. It's waving at me. So organism B is this name right here. So that's how it used a dichotomous key to identify the names of an unknown organism. And then we have phylogenetic trees, also known as cladograms. They are used to show the evolutionary relationships among a group of organisms. So here we do have the phylogenetic tree of life, so literally all living things. You will probably be given a tree that's going to be a little bit more specific, but same style question. So they want to know, are fungi more closely related to an animal or to a slime mold? So if we go over, first of all, we have to find fungi. Here they are. So are they more closely related? You see, here's animal, and then here's slime mold. Well, the fungi is physically closer to the animal on this tree, right? And the slime mold is one step away. So fungi are more closely related to animals than slime molds. And I know this because the fungi and the animals, they are closer together on the phylogenetic tree. Okay, therefore they must share, right, a more closely. They either share a more common ancestor or they share more traits in common. But fungi are more closely related to animals because they are closer together on the tree compared with the fungi and the slime mold. Okay. All right. So I told you this was a long one, but we made it through all of goal 3 for genetics and evolution. So hopefully that was a good recap of everything for right, all of these lessons. If you still have any questions or concerns, just let me know.